Hello everyone, welcome to the CeADAR Tech Talk series. So for today, we have a talk from Professor Denis. Professor Denis is Professor Clinical Bioinformatics in the School of Medicine, UCD, and today's Professor Denis is going to talk about research on peptide with application in drug development. So I hand it over to you, Denis.
Great. Yeah, I'm going to talk really fairly specifically about my own research interests, which is a little bit selfish, but it's what I know about. And so if I was to talk about the other stuff, I might just be misleading you. So here goes. I'm going to share my screen and hopefully this is going to work. It worked a minute ago. Share screen. And hang on. Talk to CeADAR. Here we go. And so I think you should now be able to see my screen. Can you see that all right, Quan?
Yeah.
Perfect, yeah. View, present mode. And that's it. Okay, so I'll get going. So that's the title of my talk. And I think the reason Quan asked me to come and talk to you, as you saw recently, we published a paper, and this was published in a journal called Science Signaling. And what we were looking at was protein motifs, which were identified from some computational analysis of looking at the SARS-CoV-2 protein spike in particular and its interactors in the human cell. I'm trying to understand exactly how do some of those interactions maybe work. So just to describe this paper and how it came about was it was initiated by Toby Gibson in Heidelberg and his research group. And then who else joined in was Friedrich Rittmann, who works in the pharma industry, Lucia Chemes, who's based in Buenos Aires, and myself got sucked into this project. And what we were studying was SARS-CoV-2. So here's a little picture of SARS-CoV-2. This is this tiny little thing, which is just a little molecular machine designed to infect cells, take it over, spread some nasty information, and make more of itself and get out again. So on the inside, you have a nucleoprotein that binds around the nucleic acid. So the inherited material here is RNA, because you might have heard that DNA makes RNA makes protein, but this particular virus has dispensed with the DNA step altogether, didn't need the middleman. So it's got an RNA genome, single-stranded RNA here, and then this nucleoprotein bounds with. And then the other major structural proteins are the big spike protein, which people probably heard of because you hear about mutations in it. And there's one or two other proteins. There's an envelope and a membrane protein here, much smaller proteins that are on the surface. And these little wiggly fellas here, they're phospholipids. That's like a standard bilayer membrane like you'd have on the outside of all your cells. And the virus has those as well. So it's got these lipid groups making a membrane, but lots of these proteins embedded crossing that membrane. So that's your basic structure of the virus. So the questions we had were simple enough is, how is this virus getting into us? What's it doing? Are there bits of it that we could understand better to help us learn to stop the virus from doing the things that it does? So here's one way of looking at a SARS virus. Sorry, I didn't have a picture of SARS-CoV-2, but it's pretty similar. So the RNA sequence can be represented by that string of a line, which then is ended in lots of A's, and that's the genomic RNA. And then superimposed on that, an image here is of the protein coding regions for each protein. So it looks like there's only 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 14 proteins there. But in fact, this protein here, after it's made, is chopped up to make lots of other proteins. So in fact, there's quite a few separate proteins being made. You might remember when I showed you the picture earlier, I showed you only about 5 proteins, which is the spike one, these 2 here in the membrane, and the one in the nuclear protein. So that's the spike one. There's the envelope and membrane ones that are in the membrane, and there's the nuclear protein. But then there's all these extra reading frames, so proteins, which do different jobs, like they might be proteases that help chop up these proteins into making smaller proteins. They might be polymerases, etc., things that make more copies of the RNA, various proteins that are useful to the RNA in making more copies of itself. So this is a masterpiece of compressing information into a very tight space. You've got this genome, and it's so restricted for space that some of these proteins actually overlap other proteins in a different reading frame, which is kind of bizarre. and weird, because you might recall that the genetic code is a triplet code that reads every 3 bases to make an amino acid, but it can frame shift 1, then you'll make an entirely different protein. So that sort of stuff is fascinating when you start looking at these viruses. So one way of thinking about this protein is, well, let's just identify all this viruses, let's identify all the proteins, look at the sequences and look in patterns in the sequences. And that's certainly one way to go about trying to understand it. Maybe make changes, make mutations, see what effect they have when we put them into cell assays. Another way of thinking about it is saying, well, of these proteins, what do they interact with? What do they come in contact with? Do they alter the behavior of other proteins by binding to them? Do they come together to make larger machines? What do all these interactions mean? So here's just a really simple schematic of a few of the interactions of viral proteins. And so up here is the spike protein that I showed you earlier. And so you can see here's a bunch of host proteins, which are kind of relevant to what I'm going to talk about later. These 2 proteins, TMPRSS2 and furin, they put little scissors beside those because those proteins are involved in actually cleaving the spike protein to make it activated. ACE2 is a receptor on our cells, which the spike protein seems to bind to. And so it's very important for entry of the virus. And then integrin is postulated to also be involved in binding. I'll talk about that a bit later, and neuropilin also. Now, what is in common between all these green proteins that have been identified as SARS-2 interactors with this extracellular spike region is that pretty much all 4 of them here have very short regions of the protein. And we call these regions short linear motifs because they're just a little simple string of the protein sequence that has a kind of independent function, like the function of being able to be cleaved by these proteases or the function of being able to bind to one of these proteins like integrin. So one of our goals is to try and make sense of these interactions that we see among the viral proteins with each other. Like, for example, these 2 viral proteins interact with each other, or between the viral proteins and host proteins, and see can we find short regions that we can maybe develop drugs that target that short region interaction. If you go to a database of protein interactions, there's one developed at EBI. It has many, many interactions among many proteins, all the experimental evidence collated together. And when we look at the coronavirus interactions, there's over 7,000 known interactions of coronaviral proteins of some kind or other. So there's quite a large database of interesting things to mine there for information. Okay, next slide. So this is really reiterating what I was saying there. Why do we want to identify what parts are doing what? I think it's kind of useful because if you know what the proteins are doing, if you've got particular mutations in particular parts of the protein, you might be able to make some predictions about what the functional effects of those might be. Up to now, people have mainly been focusing on the ability of those proteins to be recognized by our immune system, our specific immune system, which develops antibodies, and that's of critical public health importance. But you might also be interested in what mutations might also alter the function of the virus to alter the way that it presents in the disease process. And then a second reason why it might be important is we might be able to develop drugs that target those subfunctions, and that's sort of a particular goal of mine. It's a tricky thing to do, but it's a very interesting topic to go for. So some of these parts, they're going to interact between the spike protein and the receptor binding domain. They're not necessarily going to just be a linear interaction of just a small segment. They often might be multiple parts of the virus coming into contact with the viral protein, coming into contact with the host protein, like that receptor that I mentioned called ACE2. But then, as I was saying, some parts function as these short linear segments of the protein sequence. So we call these short linear motifs in proteins. Also, an acronym that's often used is SLIMS. So Rich Edwards, who was working in my group a few years ago, he used to love acronyms. So he came up with this one, and it seems to have stuck. So just to remind you, proteins are strings of 20 amino acids, which you can represent by alphabet letters. So an example of this would be a very short 3 amino acid string, which would be like a little subsequence of the protein. And the way that's represented in this single letter code is RGD. And what that stands for is R is an amino acid called arginine, G is an amino acid called glycine, and D is an amino acid called aspartic acid. So this is a positively charged arginine here, a neutral and very small glycine, and then a negatively charged aspartic acid amino acid here. And so those particular charge properties of these amino acids are very particular and important to the binding that they do. And what they're found in, among other proteins, they're found in a clotting protein called fibrinogen, which is when you form a fibrin clot, the fibrin is a major protein in your clot. And this protein, we know, fibrinogen, we know that it binds to a clotting cell type called platelets. And on that clotting cell type called platelets, there's a protein called an integrin, which specifically binds to this RGD motif. And drugs have been made out of this RGD motif. And it seems what happens when you make a peptide of RGD or a drug that makes it even more powerful, it slots into the place where the RGD should bind on the integrin receptor, and it blocks it, and that will stop other things from binding to it. So thus the fibrinogen, for example, cannot bind to the platelet. So here's a picture of the drug. Interesting question for any of you who are wondering what's going on here. This is the chemical structure of the drug, where anything where there's just a join in the thing means a carbon, and this is the structure. And this here is the amino acid arginine, that's glycine, and that's aspartic acid there. So you can see what there is here. So it's a peptide bond between each of these groups. There's an arginine here with a peptide bond here. That's where you get an acidic group and an amino group come to covalently joined. And then after another peptide bond here, you've got the D here. And then this whole thing has been cyclized to make it much more stable and prevent it being degraded. And this is a drug called selengitide that's used clinically. So then the real question is, if we have other of these short motifs, can we make drugs out of them? And can we sort of start to target their signaling in some way? So a good place to start is, well, let's go to a database of these short linear motifs that are found in eukaryotes. That's the Eukaryotic linear motif database. And you can see in this database, there's about 290 different kinds of these motifs have been identified. And then experimentally proven, there's a few thousand on the database, but we know there's tens of thousands of these motifs in the human proteome. It's just that we haven't experimentally identified them all. So what these motifs do is lots of different jobs here. They can bind to proteins. They can target the proteins to go into certain parts of the cell, like to go into the nucleus at the very center of the cell where the DNA is kept. They can cleave, they can be involved in cleavage. So you could have like for example, that furin protease involved, implicated in possible cleavage of SARS-CoV-2, cleaves at a motif which is R something, R kind of, it loves to cleave somewhere like that. And there's a bit more specificity there. So you have these kind of motifs for saying at which sites that the proteins are going to get chopped up. And then degradation, if you have certain motifs, proteins get targeted off for degradation. They might, for example, and those might also be involved with modification. So if you make a modification on a particular part of the protein, that might target it for degradation or target it for movement into the cell. So you sometimes have combinations of these motifs working together. So if you modify something, it then might activate its ability to bind ligands or to target things or to be degraded. So it's an interesting sort of network of short motifs in proteins that do things. And because they're very simple, like that RGD, they're really just short, regular expressions of 2 to 8 amino acids. We can use very simple computer search tools, regular expression search tools, to find them. So here is my puzzle today for all the non-biologists in the audience. I'm going to force you to do a bit of sequence analysis. So anyone who's a non-biologist, I'm going to ask you a question. I'm going to tell you a few things about SARS-CoV-2. One is, what's going on here? On the very top line, we've got the spike protein from SARS-CoV-2, and then just below it, we have the old SARS one. So you can see those two top lines are pretty similar because we've lined up the amino acids. So you might remember that viruses undergo mutations, and some of those mutations are changes in the amino acid sequence. So if you go along here, you can see that starts MFV, but the old SARS starts MFI. So already, before you even get to the third amino acid in this, you're getting a change. So a mutation has happened there. But other kinds of mutations can occur. Look, if you go a little further along SARS, it's deleted 2 residues there. So it goes LTT something something N, and then in the SARS, of the old SARS, it has those there. So we don't quite know if it's an insertion or a deletion, but it looks more like a deletion possibly in the SARS-CoV-2, because our other proteins share that non-deleted bit. So it's quite a challenge computationally to line up all these sequences. So Quan can tell you all about that. He used to work with Des Higgins, who is a world expert in lining up the sequences, the algorithms you need to do all that. I'm not going to talk about that today, but I'm just going to present you with a bit of this data, so you get a feeling for the sort of scale of the data. This is spike protein, is not an enormous protein, but it's pretty long, because after you put in all the gaps that allow for all the insertions and deletions during evolution, and the changes that occur, you can see it's a lot of change happening. So it's quite a lot of information to absorb. Oh, what the hell's going on here? How do I follow all these patterns? And so as you can see, I'm just jumping along to position 1,000 then in that alignment. And this is the second part of the spike protein. So I'm not going to tell you too much about this, but just to sort of get you addicted to sequence analysis, because all this data is free. You can just go and stare at it. You can get alignments. There are great tools out there. Just to start to get you addicted to this is to see if you can identify where is the region that the spike protein of SARS-CoV-2 actually enters into the membrane and goes through to the other side. So I'll give you a hint that most of the protein is on the outside of the cell and only a short bit on the inside, and that the bit that crosses the membrane is going to be full of amino acids that are hydrophobic. And in the Clustal coloring method that's used here, hydrophobic amino acids are colored in blue. That's leucine, isoleucine, valine. So anyone want to sort of hazard a guess where they think the transmembrane region is, the bit of this protein that actually crosses the membrane? So this is really just a color pattern recognition problem here. So which part of this sequence would you reckon is the bit that crosses the membrane? On which line? Is it in the first, second, third, or fourth line of this slide?
I think it's the 4th line, right?
4th line.
Yeah, the 4th line, because you've got this big patch of blue. And another thing that I happen to know about transmembrane regions, they're often about, say, 18 amino acids long. So it looks about the right length to be it. So that's exactly it. So already you're doing sequence analysis. You're looking for patterns. You're trying to make sense of them. So any biologists in the audience, there's a bunch of cysteines here just on the inside of the cytoplasmic tail. Any suggestions what that might be doing here? No ideas?
I'm not an expert in this.
No, there's no expert in this. Right, well, I thought
maybe stabilize the protein.
Sorry?
Maybe cysteine bonds to stabilize the protein.
Yeah, that's what it could be. That's what I thought when I saw it first. But actually, when I went up reading literature, it turns out that these proteins have quite a lot of palmitoylation modification, and which often happens by attaching a lipid group to the cysteine. So my suspicion is that what these things are all doing is that they're involved in the addition of the lipid tails, extra lipids to this. And this is right beside the membrane. So it's probably really locking this whole region into the membrane, because those lipid tails are very fatty and they'll love to embed in the membrane. So I think there's a very interesting structure being made here by the virus, where I think that's fascinating, but there's very little literature done it, and I really don't understand what's going on. So already we've got interesting things, interesting patterns happening that we don't really understand just by eyeballing the sequence, which I think is fun to do. So let's get back to our motifs, because I wasn't asking you to look for motifs in that sequence because the font was not so easy to read. But here's from our science signaling paper. We found a region of the SARS-CoV-2 protein. And then, to be honest, we weren't the first people to spot this. There's a few papers came out, identified this, but maybe they didn't analyze it in such detail. But essentially what we found was that remember that RGD that I said binds to a protein called integrin? We noticed this motif was here. So we were wondering, well, is that just by chance, or is that functional? So it's a little bit hard to say, because it's not enormously conserved. Often in biology, we say if things are conserved, say that cysteine there is quite conserved. It's in all the different species here. OC43 is a virus that causes a common cold, by the way, just in case you're wondering what that one is. Various other batty things in there. And then the old SARS and the new SARS are in this little picture. But what we saw was that in the OC43, there was an NGR motif. And what we thought was really interesting, almost overlapping at a very similar position. And we thought this was very interesting because NGR is known to be capable of binding to the integrins in the same way that the RGD binds at the same site. So we kind of thought this provides some evidence that it's more likely to be functionally important. I'm still waiting for really convincing experimental evidence that the RGD binding to integrin actually matters for the biology of the organism, because although we think the evidence pushes towards functionality, we don't really have clear proof. But it certainly is worth experimentally testing peptide drug reagents that bear the RGD motif. Even if they're not critical, they might help hone the virus towards regions that are rich in the integrins. This is a way of representing the sequences when we line them all up, and this is all done in the cluster program. Another way of representing the similarity of the sequence is to use an evolutionary tree. So that's down here below here. So as you can see, sorry, this is the spike protein, that's the full protein. This is the beat region here that binds to various receptors. This is the one that's most important in binding to the ACE2 binding region, and then this might be a region that's binding to integrin. So it's kind of interesting to see this effect. And here's a structural representation of how the spike protein is all folded together, and it's exposing this RGD, this tripeptide motif represented here in a kind of molecular summary graphic format as sticking out. These lines here are beta sheets, and these are alpha helices. These twisty bits are different protein structure forms that are detectable in the structure. But another way of representing the information is as an evolutionary tree. So all this does is it simply groups the two proteins that look most similar on top together, and etc. And it just tries to create a tree based on the similarities. So that's straightforward enough. And then when we start to look at that ACE protein, which we know binds to SARS-CoV-2, and here again, you might see a big hydrophobic region here. You're getting used to this already. These are very blue, and this is the bit that spans the membrane. But beyond that, then we got the bit that's inside the cell. So on the outside of our cells is most of the ACE2 protein, all this very large part. But on the inside of the cell, there's a short region that sticks into the inside of the cell called the cytoplasm region of the cell. And when Toby and his group all looked for motifs, they found a whole series of interesting motifs in this region. And these were very suggestive and very interesting. And what was most interesting are the ones which might be important in trafficking the virus around the cell. So endocytosis is a particular process where you've got a cell membrane and the cell gulps things in from outside by taking them in, by forming a kind of a little loop around them and then just closing in and bringing in a bubble of material from the outside. And that's one way that viruses can get in. And then a second thing that's very important for trafficking the virus around the cell is a process that's called autophagy. And this is a process where the cell gulps parts of itself to break down and eat up parts of itself. So for example, if you've got old parts of the cell, organelles or proteins, they might get autophage. It just means self-eating essentially. So there's a process involved in that. And this seems to be, both of these things seem to be very important for SARS-CoV-2 in how does it go about the job of getting itself into these vesicles where it can be safe from all the antiviral mechanisms that the cell has to try and kill off the virus. And so coronavirus are very good at getting into that space and protecting themselves from the host machinery. So the guys found these motifs, which was very interesting and very, very fascinating. And then the question really was, well, do they actually bind the way they're meant to? Because these are just computational predictions. The regular expressions are relatively short. How could they have just occurred by chance? And so to answer that question, oh yeah, sorry, that's just this is just a visual summary then in terms of all these motifs that are identified and their potential roles on the inside of the cell. From the ACE2, we also looked at integrin motifs as well on the integrins. And here, do you remember that RGD motif that I was saying is on the spike protein? So that's it just visualized here, how it combines to the integrin here. That's our thinking about that. So this is just an overall schematic saying these are all the points in the talking between SARS-CoV-2 and the host cell where we think these motifs are important. So these computer predictions are great, but how do we go about telling if they're real? And so some friends followed that up. Ylva Iversen in Sweden did the experiments where she took those peptide regions from the ACE2 protein and tested, do they really bind to the domains where they're meant to be binding, where that database of motifs said, this is the motif for binding that protein. She and her team then went and tested that, and they found indeed that it did. And some of the technology that she uses is really fascinating. So this is a bit of work that Norman Davey, who used to work in UCD with us, he's now based in London, but he did a lot of work with Ylva. And they've done some fascinating work to try and create a kind of combined computational experimental pipeline for identifying the most relevant protein regions, making little peptides out of them, and then testing them. But instead of doing what I'm going to describe in my projects, which is only testing, 20 to 60 peptides, they can test thousands of them. And they use a thing called phage display, where you get a little bacteriophage, which is a virus of bacteria, and you engineer it by molecular biology to express the peptide on the surface. So this is a technique that's been used for many years to present random combinatorial peptides. But the beauty of what Norman and Ylva and Philip Kim do is that they take peptide sequences that we know about from biology. So they take the entire set of human proteins, they use a machine learning predictor to predict the disordered regions that aren't going to be globular. So all the line bits that don't seem to form a structure, they want those because they're the ones that we think are important in motif interactions. So they get rid of all the globular bits that seem to form three-dimensional structures and are less likely to have the motifs in them. And they just focus on these other bits using a machine learning predictor here to identify those regions. And then they go through a protocol of processing all those sequences in the computer. And then finally, they have sort of some really classic, nice molecular biology to just high throughput synthesize all of those peptides and present them on these phages. So you get a massive library of these viruses, all of which have a different peptide on the surface. And inside each virus, you got the nucleotide sequence encoding them. So then if you make all these peptides and you bind them to something, like a protein you care about, they stick. And then you sequence the DNA out of the ones that stick. And from that, you can identify the sequences of the peptides that you've got. So it's a really classy technology. This is just an example of how they've found particular proteins, which are the binders. And then this blind method of discovering the interactors has identified the interactors. And when they go and look at the motif that's discovered, this really gives you a very unbiased way of defining your motifs because you've scanned such a large library of real biological molecules to look for the binders. So this is a really nice way of actually systematically going at this rather than just chipping away with very small bits of biochemistry. So it's a classic, fascinating technology. Now, for me, the difficulty with this technology is not that it's wonderful. It is wonderful, but it's really good for binding assays. But I don't really see a way to apply it to bioactivity assays. So we could do this to get binders to the spike protein, for example. But could we assay for biological activity with these very large libraries? I don't really see an easy way to do that at the moment. So that sort of leads me to a question, then what strategies are we using to go from these short linear motifs that I was talking about to the anti-SARS drugs? And this is where I'm at the moment. We recently got a grant funded to try and have a go at this. So it's forced me to go back in time a little bit and think about some of the things we've done in the past and what worked and what didn't. So previously we tried to screen computationally for motifs. We'd synthesize them as short protein sequences and test them in the lab. An example of one time we did that, we were working on those platelets that I mentioned earlier, those blood clotting cells. And we looked in the bits of those proteins that are stuck in the membrane. We just looked on the bits that are inside the cell, and we tried to identify the very interesting SLIMs in those, the short motifs, and we tested them experimentally. I won't go into the details of this. It was a fairly convoluted logic, but we threw up some really interesting findings, I think. But the difficulty and the challenge for us was really probably in further experimental follow-up beyond the initial sort of validation. And from that point of view, in terms of further validation, we were really interested in digging down to peptides which had effects at very small concentrations. And so Kevin O'Brien set out doing a very ambitious screen. He took all the proteins in this set. He compiled a set of proteins which interacts with integrins in the platelet, with the platelet integrins. So there's that one there is integrin, and that one there is integrin. They're two integrins because they come together. Two different proteins come together to make an integrin complex. And then all these are the interactors that interact with the integrin or with the components of the integrin. So this is kind of horrible hairball network representation. There's been a bit of an attempt by Kevin's software here to make sense of that by putting in some kind of order, like the things that are in the membrane or outside the cell there, and then the things that are adapters and things that are kinases and things that are phosphatases and other here, and actin regulators here. But to be honest, you know, this layering is not even a natural physical layering. So it's just some way of trying to reduce the information down. And so Kevin identified peptides in all the blue proteins and synthesized peptides from those proteins, and then tested them in the laboratory. So this is a pretty ambitious thing for a computer scientist to come in for his PhD and do, to go from all that design to the experimental testing. So it's a real, it was a pretty much a triumph to get all that work done, both the computational and the experimental. And then I think in red and green, we've identified the ones which were interesting from an activity point of view. So this is just a table just to say this is what peptides are, sequences of amino acids. You can't see them what they are, but you can see some of them are a bit shorter, some of them are a bit longer. Some of them we put a PAL at the front. Do you remember I mentioned in the spike protein that there are those lipids that are attached? What we find when we add lipids to peptides, it helps them embed in membrane and get into the cell, and then maybe dangle from the membrane because they remain stuck in the membrane. And that's kind of exactly where we wanted these motifs to be. And then for some of the motifs that weren't in membrane proteins, we added a different thing, a thing called TAT, which is a cell-penetrating peptide of about 11 amino acids, mostly positive charges. It's derived from a virus protein, but we know that it helps get the peptides into cells, which is where we wanted them to act. And then for the computer prediction, how do we select these? We use two different softwares, a machine learning predictor called SlimPred. And that was based on, if you remember, I showed you a database called the Elm database, which listed all the SLIMs, and it gave all the instances. So essentially, it was as simple as this. We trained it with all those instances to see, could we end up with useful predictions of what sequences are SLIMs. And we also used another method, which was based more on evolutionary dynamics and evolutionary conservation, because often the bits that are kept in evolution are more important. And that was, so these two softwares were applied. And then we also tested for, do these peptides interact with each other in a synergistic way, because we were intrigued by that. I won't go into the definition of synergy, but we spent some time defining our definition and our experimental strategy here. And we've a paper in PLOS Computational Biology about that strategy. But the essence here really was you're just looking at all these peptides and not just all pairwise combinations to see what effect they have. And we also made combinations where they weren't just added separately to the test tube before we see what effect they have on platelets. We also conjugated them together as one peptide chemically joined to another. So we tried different strategies. One thing we noted from our findings was that the vinculin binding peptide seems to co-localize in cells away from the membrane. So we actually took photographs of cells here where the vinculin, when we add the peptide, seems to, instead of being out at the cell membrane, seems to be sort of going to these funny inclusion bodies, suggesting it's affecting the trafficking of the vinculin protein. And then we also noted that if we put that, remember I said there was a positive charged peptide that we used to help the peptide that we were interested in to get into the cells. Well, we had a choice. We could either add it to the left-hand side or the right-hand side of the peptide. And what we found with this vinculin binding peptide was if we added it at this side of the peptide, it looked like it was, when we did structural modeling. So this is a model of, this is a model of the protein, a three-dimensional model of vinculin. And this here is the binding site where the peptide binds. There's a real groove, a cleft where a peptide can slot in there. And you can see it's in this alpha helical kind of formation that can slot into that space. So when we compared, when Kevin did the structural modeling and he put the TAT at one end or the other, he noticed these structural models were very different. In this model here, the TAT is very doesn't form a very strict position. It's wobbling all over the place. It's not very stable. In this one here, it's much more stabilized because there's some kind of charge complementarity of the positive charge of the TAT with the protein, the vinculin protein. So where you add these and how you add these cell penetrating peptides can have a big effect on the efficiency of binding of peptides. So that's one of the things that we learned from that study. And we also found that the synergistic effects really weren't all that strong within the network, which I was disappointed by, but it was a reasonable biological observation. And then the synergy that we did notice was really, for me, two of these peptides are right beside each other in the sequence. When we made them as a single large, bigger peptide, that was much more potent. And so it was more like it was just affecting that one particular binding thing in a much more potent way. And I suppose there were some lessons from that. Should we be screening with larger peptides to start? Another piece of work we've done in the past is to screen for interactors. I think I'm using up a lot of time here, Kwan, so I'll probably try and speed up a little bit.
I think we have talked for like 40-50 minutes, so until like 11:55.
All right, okay, yeah. Oh, sure, I'll keep going for a little bit.
Yeah.
Another thing we've done is we've taken another approach to these motifs, because if you think about it, these motifs, you might remember when I showed you that database, I was showing you there were maybe 260 known kinds of these motifs, but in fact there were a much larger number of instances. In other words, the same motifs are used again and again and again, and that's because a single protein, which has a slim binding domain, a domain that binds these peptides, can bind to many proteins that all have this short motif. So on the right here is an example of this, is a protein called PDE6D, and this shows you the interaction network of all the proteins in a database like that intact database I showed you earlier that interacts in humans with PDE6D. So you can see there's some self interactions, but then there's interactions of PDE6D with all these other proteins. One or two of these proteins actually interact with each other as well. But that's the kind of picture of the set of interactions. And then when you go and look at this network, what you find is those green proteins are ones where the motif that PDE6D binds to at the very end of the proteins, at the very end of these proteins in green, they have the motif. Now, these other proteins in blue don't have the motif. So the interaction between this protein and this protein must be by some other mechanism. So then when you have, this is the knowledge that we gained, we actually identified this pattern not from experimental evidence, but from computational analysis. And how we went about that was we just said, let's take all the human proteins like PDE6D and make a data set of all their interactors. So make a file with all their sequences in it, and then ask the computer program, look for some regular expressions that are in common, that are enriched in this data set. So there's a few different tools out there you could have done this with, but what we found was that none of them were working very well. So we ended up with quite a good tool that Rich Edwards worked on, where we looked for the overexpression of enrichments. And then the things that Rich really did in terms of advance this was he found a good way of thinking about all the possible regular expressions without going crazy. So he maybe limited from 2 to 8 amino acids length, allowed for certain kinds of gaps, allowed for certain kinds of ambiguity, because some amino acids are quite similar, and we're interested in motifs which are ambiguous at certain positions. Then we did a correction for evolutionary relatedness, because many proteins in a data set like this will share regions that are not interesting from a short motif point of view, because they are homologous proteins that have a very large region of similarity. So we wanted to correct for that, and we found a way to correct for that. So these RL proteins here are all related to each other. They all have similar sequences. So therefore, you want to correct for that, because otherwise you'll just end up identifying the similarities of the RL protein that you could find much better with a protein designed for finding global similarity instead of just local tiny motif similarity. And then the next step was calculating the probability, and this was done really nicely by Norman and Rich, to how to think about the probabilities of these things. So we end up with software that's really good. You just run a set of proteins on it and you get a probability that is pretty actually robust. And so this software then on this data set returns back the most highly ranked and significant motif is this C something, IRL. Remember I said they could be ambiguous, something, and dollar is the standard regular expression term meaning end of sequence. So that's that piece of software, and they went on together to identify lots of these motifs in humans. So one of the things then to think about also is quite moving away from that sequence bioinformatics, which is all about the linear sequences, to the structural bioinformatics, and this is all at the molecular modeling. And in this case, we're very interested in these cleavage motifs that are in SARS-CoV-2 that are for these enzymes, because we know that the virus is going to be activated by these enzymes. It would be very nice if we could make peptides which are protected in some ways and then activated at the site they're needed by these self-same enzymes. So we're very interested in making reagents that do this, that are activated by this. So from this point of view, Ajay Pal, who's just started working with me 2 weeks ago on this, he's done some work here of homology modeling of that key protease. So the three-dimensional structure of many proteins is evaluated experimentally. This particular one doesn't have a structure available, but he's able to come up with a good homology model. And when he looked at this binding site of the peptide, it seems to be pretty similar across many of the different enzymes, regardless of whether they're binding to a ligand or not. So this region seems to be quite a constant, held in a certain fixed way, so that you can then use software called docking software. And some of this software itself has machine learning elements to it. Kwan, I'm sure you'd be interested in that. There are different strategies that are used by different softwares. So the docking of this peptide region to so in fact, Ajay did this in a fairly crude way. He just said, look, dock anywhere in the protein. And nicely enough, the SARS-CoV-2 peptide at the critical cleavage site, when we took that peptide, it actually docked exactly to the correct binding site. So the computer program was able to reproduce the correct kind of binding conformation. And so we're very interested in some of these interactions, because this long arginine here is very important for cleavage, and it looks like it's got this nice charge complementarity in the structure. So when we're synthesizing cyclic peptides, we're going to be testing them against this model. So what Ajay has here is a really nice model for us to go and computationally screen any compounds that cyclic peptides we design to make sure that they look cleavable, because there's no point in us making things which aren't going to be able to adopt the conformation that the enzyme can cleave. So that's where we're at with that project. We've got this model for how we might test for cleavage. We've also got the parts list of done from SLIM analysis in the Science Signaling and paper, and some other analyses I've done. And so then what we're going to try and do is we're going to try and look at that part list. So we've got the peptide activation ones, those motifs for making sure they're cleaved correctly. Then things like the RGD integrin or the neuropilin motif. I didn't go into this one, but it's, you know, similar kind of story to the integrin one. Can we add these to the peptides? And then can we take peptide regions or SLIM regions from the viruses that are going to disrupt the viral proteins? And we know there's a really good reagent against HIV which can do this. So we're interested in sort of modeling in a similar way. And now we've got to combine these and design them and synthesize them in a chemistry lab. So that's where Mark Devasel comes in. He's a chemist up in the College of Surgeons, and he's going to do that work. And then once they're synthesized, we'll test them for their activity. And then the most important test of all is in a level 3 containment lab. So Virginie Gauthier in UCD has a level 3 containment lab where she already has the screens up and running for SARS-CoV-2. So she's got live and dangerous SARS-CoV-2 virus in her lab. And they've got, you know, quite strict protocols for the to ensure it's safe, and it's rather unpleasant working conditions, to be honest, but that they're committed to doing that work. And there we're going to be able to screen these peptides for their bioactivity, for whether or not they can prevent the entry of the virus into cells. And those cells will be cells grown in what's called a tissue culture, where you take human cells or animal cells and you're growing them in the lab, and then you're seeing, then you grow the virus on the cells, and then you're seeing if adding the peptide will stop the virus from entering or growing in the cells. So that's the summary there. We're going to design the peptides, check if the human cells are going to do the activation, make some further design choices, and then do the tests to find what peptides inhibit them. And then hopefully we'll get some leads. And from those leads, we're going to just sort of really try and push them forward, combining both experimental and computational approaches. So that's sort of where we're at. And you can see there's a lot of things in this in terms of the overall motif approaches that I've described in my overall talk, which might not be included here, but we think this is going to be the strategy that makes sense over this sort of scale of work, because we're trying to complete this work all within a single year. So it's slightly pressurized. It's this rapid response, and I've never tried to do research at such speed before, and I don't know if that's going to be a good thing or a bad thing, but I'll let you know in a year's time. So thanks very much, Kwan, and everyone for listening, and I'll stop my sharing now.